The overall goal of the following experiment is to segment features of interest from complex 3D electron microscopy data sets of cells and tissues for analysis of their 3D organization. This is achieved by collecting a data set consisting of individual electron microscopy images. The raw 2D data is then reconstructed into a 3D volume and filtered to reduce noise and enhance the features of interest. As a second step, the data's objective and subjective characteristics are assessed to inform the best choice of method for segmentation. Next, either manual abstracted model generation, manual tracing of features of interest, automated density-based segmentation, or custom-tailored automated segmentation are performed to extract the features of interest. The results show final 3D segmented models of features of interest based on triaging image characteristics and personal aims to choose the optimal segmentation approach. A comparison between the different approaches for the different data set will help select the correct segmentation strategy.
We demonstrate different methods for the extraction of features of interest. Given the complexity of subcellular electron microscopy volumes, each approach has its advantages and limitations.
Generally, individuals new to segmentation may struggle because identifying the best approach for different data sets isn't always clear.
Along with graduate student Wen-Ting Tsai, demonstrating the procedure will be Purbasha Sarkar, a postdoc in my laboratory, Ahmed Hassan, a research associate in my laboratory, and Joaquin Correa, a computer systems engineer.